thank you guys all for joining me today. Thank you, Mike and Jen, for those introductions. I'm super excited to share this talk with you guys today. It's called Trustworthy, Scientific Inquiry and Design with Machine Learning, a Theory and Wet Lab Guided Tour of Trust Issues. Okay, so I'm gonna start with a game. Here are two protein structures. One of them is an experimental structure derived from wet lab. The other is an AlphaFold prediction. Can anyone guess which one is which?
I know.
Yes. Go.
Well, the AlphaFold prediction is that terrible thing on the right.
Okay. Any other guesses? I know there are people here with the expertise to make.
Oh, actually, I don't know if any expertise. I just was an author on the paper, so no expertise needed.
All right, so turns out the one on the left is the AlphaFold prediction.
Oh no! You lied to me.
And the point of this is that it's not entirely straightforward to tell, even though the AlphaFold one gets some important parts wrong. And so the high-level motivation of my work is that we, as a scientific community, are increasingly using machine learning as an inexpensive, high-throughput proxy for scientific experiments. And indeed, predictions from systems like AlphaFold can dramatically accelerate the process of science by serving as proxies that are less expensive, less resource and time consuming than gold standard experiments. So machine learning systems have been used as proxies in this way for both scientific inquiry, and by that I mean the discovery and quantification of scientific phenomena, as well as the design of novel objects, including designing incompressible materials, small molecules, and enzymes, and fluorescent proteins, a whole bunch of other applications here. However, there's a problem, and that's that predictions contain errors, and that's something we all understand. So if I draw a scientific conclusion based on looking at a bunch of AlphaFold predicted structures, how do I know that that conclusion wouldn't have been different if I'd been looking at the experimental structures instead? Or if I design a new protein therapeutic based on some crazy protein language model or some other machine learning predictive model of binding affinity, how do I know that designed protein will actually behave the way I want instead of the model just being kind of wrong? So to address this, I'm going to share 3 stories from my PhD tackling this question. How can we use machine learning predictions to do trustworthy scientific inquiry and design? So stylistically, this talk is not going to be about ways to train particular models or architectures, but rather, given that you have a promising predictive model for some domain, no matter what that model looks like, how can you then use its predictions to do science in a reliable way? Okay, so this question has 2 parts. There's the scientific inquiry, there's the design. So I'll address this first sub-question first. So I'm going to share work from a recent preprint called Prediction-Powered Inference. This was a super fun project with Anastasia, Steven, Tiana, and Mike.
What's on your hair, by the way?
Oh, that's snow.
That is snow.
Yeah. I realize it may look like other things, but
It's like volcano ash.
So to start this, I'm going to start with a motivating story about proteins. So proteins are a sequence of amino acids that fold into a 3D structure. And studying features of these sequences or features of their structures can give insights into protein function, disease mechanisms, and medicine. And in 2004, biologists made an important observation that a particular sequence feature called a post-translational modification seems to occur very frequently with a particular structural feature called an intrinsically disordered region. And this is actually just regions of a protein that don't have a fixed 3D structure, they can kind of flop around. So details aside, this was actually a very insightful observation that would go on to motivate many lines of research in biology. But it turns out that back in 2004, they couldn't actually quantify this association between these 2 features. They kind of made this observation heuristically, but they couldn't quantify it because there just weren't enough high quality experimental structures at the time. Fast forward almost 20 years, now we have AlphaFold predicted structures for hundreds of millions of protein sequences. And last year, a study came out that used these really abundant predictions to finally quantify this association. And they did this by computing an odds ratio between the presence of these 2 features. Okay, now the twist here is that they weren't actually looking at the structural features. They were looking at predicted structural features based on the AlphaFold predicted structures. So this story points to a bigger story happening all over science right now, which is that predictions are being used for scientific inquiry. And we see this not just in proteomics, but also in studying disease, regulatory DNA, the demographics of patient phenotypes, and even beyond biology to studying human welfare more broadly. And generally, it makes sense to be doing this. Machine learning predictions are super cheap. They're basically instantaneous, very easy to get, whereas gold standard experiments are very expensive, time-consuming, require highly trained postdocs or grad students. So this is very cool. Despite this, it should make you a little bit uncomfortable when predictions are used to draw scientific conclusions. And the reason is just that we understand that they're not the same quality as gold standard experiments. So for example, the paper we just saw that was using AlphaFold predictions to compute an odds ratio, if it then reports an odds ratio of 1.2, how do we believe that number? Like, how do you know you can trust 1.2? Is it really 1.2? So to tackle this, we proposed prediction-powered inference. This is a framework for provably valid statistical inference when we use predictions as if it were data. So this is a general problem setting, which is that we have some labeled data consisting of features X and a label Y. So, sorry, find my cursor. So Y is anything that you would typically want to measure with some gold standard experiment. So for example, X could be a protein sequence, Y could be the protein structure or some feature of that structure. X could be a regulatory DNA sequence, Y could be the gene expression level it causes, X could be some deep sea image, Y could be labeled species, for example. And I will sometimes call this labeled data the gold standard data. And in addition to that, we also have this much larger set of unlabeled data indicated by how giant these bars are. And these also have features X prime, but here the labels Y prime are unobserved. So for example, this X primes could be like all possible protein sequences I would have been interested in, but I don't have the structures for, or a giant database of deep sea images that haven't been annotated by an expert yet. Okay, so importantly for both of these data sets, we have predictions, F and F prime, that are made by a machine learning model. And throughout, I'm just going to assume that this model is already trained on separate data, and it's just a fixed prediction system. So we focus on a common setup in science, which is that there's some estimand that we want to know the value of. And this estimand is going to be some property of the underlying data distribution. So for example, it could be the mean value of Y, some quantile of Y. It could be regression coefficients between Y and X if we're trying to look at a relationship between things. Yeah, and just to instantiate a bit more, the mean value of Y can capture any kind of question that's asking me about the frequency or the count of some label of interest, for example. Okay, so our goal in this setting is to construct a confidence set, C alpha, that contains the true value of the estimand with guaranteed high probability, greater than 1 minus alpha for any user specified alpha. And we call this validity. So how might you go at this goal with existing tools? In what we call the classical approach, you could just use the labeled data, forget the crazy machine learning predictions. And using existing statistical tools will give you confidence sets that are valid. They have this guarantee. But you're completely losing out on the information that is contained in the predictions. So even though, for example, AlphaFold predictions are imperfect, they're still like really, really good, which is why everyone's so excited about them. So if you're just totally ignoring that, you're just tossing out evidence. So on the other extreme, in what we call the imputation approach, you could just imagine treating the predictions as if it were gold standard labels, carrying on with your day, doing what you would have done otherwise. And if you do this, existing statistical tools will give you confidence sets that are invalid because of prediction error. They won't actually necessarily contain the thing that you're trying to estimate. Okay, so prediction powered inference is a framework for using both the labeled data and the unlabeled data to get the best of both worlds. We'll get confidence sets that are provably valid while still squeezing information from the predictions to basically increase sample size. Okay, so before we see how that works, I'm going to show a quick demo using data from the paper we saw that was using AlphaFold predictions to compute an odds ratio. So for this demo, we looked at the portion of their data that did have gold standard structural features, gold standard labels for the structural feature. And on that data, we can compute the true odds ratio, which is this vertical dotted line. We can then simulate having unlabeled data by throwing out the vast majority of those labels, pretending we never saw them, and instead getting predicted structural features from AlphaFold predictions. We then use the classical approach, computation approach, and our approach to construct confidence intervals for this odds ratio. So I'm going to show all three of those intervals now. As expected, the classical one is valid. It contains the true value, but it's pretty wide due to the small amount of labeled data. On the other hand, the imputation confidence interval, while very small, totally misses the true value. So prediction powered inference, the one in green, gives us the best of both worlds again. It's valid because we're correcting for prediction error, but it's still smaller than the classical one because we're basically leveraging the predictions as extra information. All right, so how does this framework work? So on this slide, I'm just going to sketch out the high-level intuition behind prediction powered inference. So it starts with identifying what we call the rectifier. And the rectifier is the specific notion of prediction error that is relevant for your estimate of interest. So if you're trying to estimate the mean value of y versus some regression coefficient between y and x, prediction error is going to affect those two things in a different way. And that's what's kind of captured by the rectifier. So to visualize that in this diagram, this theta star on the right is the estimand that you want to be estimating. For example, it could be the mean value or expectation of y. And theta f on the left is the analogous quantity, but based on the predictions f. So for example, the expectation of f. And theta f here is generally going to be biased due to prediction error. And the rectifier, delta f, is the notion of prediction error that we need to kind of correct this prediction-based quantity and get it to the true value theta star. So in this example, the rectifier is super intuitive. It's literally just the bias, the expected difference between predictions and labels. And for other estimands, the rectifier will be different, but we give a general recipe for how to identify that rectifier for a broad class of estimands you might care about. Okay, the second step here is to construct a confidence set for the rectifier. So the rectifier is a quantity that needs to be estimated. So we use our small amount of labeled data to build a confidence set for this rectifier, which we call R. And the final step is that we form the prediction-powered confidence set represented by this blob in green by propagating theta f through all possible values of the rectifier in the rectifier's confidence set R. So this set basically contains all values of theta that result from taking this biased thing and correcting it with a high probability value of the rectifier. And the idea is that so long as this confidence set R for the rectifier is valid, then our prediction powered confidence set will also be valid. It contains theta star. And one intuitively nice thing I'll point out is that the rectifier is generally going to be small if your predictions are good. So for example, here, if your predictions f exactly equal y, then the bias is just 0. And so the result of this is that the better your predictions are, the smaller the rectifier and the smaller your resulting prediction powered confidence sets. Okay, so I'm going to make that a little bit more concrete by just briefly sketching out our main technical result, which is that this procedure we propose does give provably valid confidence sets for a broad class of estimands, and specifically for any estimand that can be written as the minimizer of a convex objective. So again, this includes lots of estimands people use in science all the time: the mean value of y, medians or quantiles, linear logistic, Poisson, other kind of generalized linear regression coefficients kind of fall into this class of things. And this is just the main theorem. Again, for any estimate that can be written as a minimizer of a convex objective, this procedure that I just sketched out produces confidence sets that are valid. So just in a little bit more detail for those who are interested, the rectifier for this class of estimates is actually the bias of the derivative of the objective. So this is the quantity that we're trying to estimate with the labeled data, and then we're going to use that estimated quantity to correct the prediction-based derivative, and then basically use that rectified derivative to identify theta star. Okay, so this is a really powerful and broadly applicable tool. Whenever you have a measurement or a gold standard label that you have a reasonable predictive model for, you can basically now use those predictions as cheap extra data to do valid science. So I'm going to finish by showing our framework in action on 2 more data sets. There's a whole bunch more included in the preprint if you're interested. So in this first one, I have a population of regulatory DNA sequences called promoters that are naturally found in yeast. And for a relatively small number of them, we have experimental measurements of the gene expression level caused by that sequence. In addition, for tens of thousands more promoters, we have predicted gene expression levels from a state-of-the-art predictive transformer model. And the goal is to estimate different quantiles of the gene expression level in this population. So on the left is the 0.5 quantile or the median. On the right is the 0.75 quantile. And the true values are these dotted orange lines. And you can see in both cases that the imputation interval, which are in orange, they're biased away from the true value because of prediction error. And our prediction powered sets in green basically correct for this error and do contain the true value, while also being smaller than the classical ones. And we see this trend of the prediction powered ones being smaller than the classical ones across a broad range of amounts of labeled data. So this is another cool example that I'll end with. This prediction powered galaxy counting. So here we have a bunch of images of galaxies from the Sloan Digital Sky Survey. And we have a small number of them labeled by expert astronomers as either being a spiral galaxy or not a spiral galaxy. We also have predictions of that spiral galaxy label for millions of additional images made by a computer vision model. And the goal is to estimate the frequency of spiral galaxies in the local universe, which is just a cool thing to say. So again, the imputation confidence interval here is biased away from the true value because it's not correcting for prediction error. Ours corrects for that error and ends up covering the true value while still being smaller than the classical ones. Okay, so as a summary, prediction powered inference is a framework for using both your labeled data and predictions to estimate scientific quantities. And you can use basically any machine learning model you want. I didn't make any assumptions about F, like how it's trained, the architecture. In fact, in theory, really, F doesn't even have to be a machine learning model. It can be any sort of computational model that provides you cheap predictions that you want to kind of unbias yourself from the error of. So importantly, we show that this procedure yields statistically valid intervals. And we also have the intuitively nice phenomenon that the better your predictions are, the smaller the rectifier, and the more prediction-powered inference allows you to basically increase your sample size for doing science. All right. So that was kind of a step toward this first part of the big question. Now, onto the second part of this question. How can we use machine learning predictions to do trustworthy design? This is a question that I've spent many years of my PhD thinking about, and Jen's laughing because I'm also banging my head about for a very long time from lots of different angles. I'm going to share 2 of those projects today, starting with an ongoing wet lab collaboration that motivates the subsequent work. So last summer, I was incredibly fortunate to intern with Ali Madani, actually here in person, and Nikhil Naik at Salesforce Research, where we collaborated with Polly Portis's lab at Stanford and James Fraser's and Margot Penny's labs at EECS. And the goal of this collaboration was to design thermostable enzymes. So that is, can we design an enzyme sequence that is more thermostable than a known enzyme that we care about, while not losing catalytic activity. So why do we want to do this? It turns out that making enzymes thermostable is a really critical step to making an enzyme useful for industry scale chemical reactions. So the production of biofuels, detergents, food, textiles, drug manufacturing, all of these things actually rely on chemical reactions that are catalyzed by very thermostable enzymes. And in fact, to convince you more, in 1993, the Nobel Prize was awarded for the discovery of a single highly thermostable form of DNA polymerase called Taq polymerase. And this is actually the discovery that paved the way for PCR to become the workhorse of biotechnology that we know it as today. So Taq polymerase was discovered in Thermus aquaticus. This is a bacteria that lives in the near boiling waters of Yellowstone hot springs, which is super cool, or super hot, I guess. Unfortunately, it turns out that most enzymes that people are interested in don't happen to exist in bacteria that thrive in near boiling water. So methods that can make any given enzyme more thermostable are actually of widespread interest. However, it's non-trivial and quite time-consuming to directly assay both catalytic activity and thermostability in the wet lab. So instead, we'd like to use predictive models of those properties trained on publicly available data so that we don't have to collect any of our own data to design these enzymes. So the question is, or one question among many, is how can we use these predictive models while accounting for the fact that we don't trust them equally everywhere in sequence space? Okay, so in a nutshell, at a very high level, we did this by introducing trust region constraints to the design algorithm. So we simply constrain the designed enzymes to regions of sequence space where we trust the models for both thermostability and catalytic activity based on our knowledge of the training distributions. And so with our method, we designed enzymes from two different enzyme families, lysozymes and acylphosphatases, which are now in the hands of our collaborators who are currently validating them in the wet lab, which always gives me anxiety. And so that's super exciting, but today I'm just going to share some preliminary teaser data. And before that, I'll note that if our method is successful, it means that we can design thermostable enzymes without any family-specific thermostability measurements, any catalytic activity measurements, any structure, either experimental or predicted or otherwise, or any manual curation of an MSA. So basically, you don't do it. That's the goal. Okay, so onto the preliminary results. There's two axes we care about, remember. We want the designed enzymes to be more thermostable than wild types, while also not losing catalytic activity. So I have initial data on one of those axes, activity, from my amazing collaborators, Mike and Craig. So the y-axis is the one of importance. This is kcat over km. It's just a measure of catalytic activity. We want this to be high. And on the x-axis is just three different wild types. So in the gray bars, these are the measurements for the wild types. And the green box plots show the activities for the design sequences. So just a high level point is it appears that our designs are at least not losing catalytic activity, which is important because you often see a trade-off between activity and thermostability among natural enzymes. So this is really encouraging. We're all, of course, waiting with bated breath for the thermostability measurements. But in the meantime, this work and this problem serve as motivation for the final paper that I'll discuss today. And so going back to this question, how can we use machine learning predictions to do trustworthy design? This last paper refined this question a bit by asking, how can we quantify the uncertainty of predictions for design? So this was a paper called Conformal Prediction under Feedback Clearance Shift for Biomolecular Design. Really great project that I was fortunate to work on with Steven, Anastasios, Jen, and Mike. So the setting in this work, which includes our ongoing work in the thermostable enzymes that I mentioned, the setting is single shot design. So in this setting, I'm interested in some protein property, which is some ground truth real value function over sequence space, which is like the x-axis And I have a single batch of sequences labeled with measurements from the wet lab. And from this single batch, without any additional labeled data, I want to design proteins with high property values. So one way I can do this is train some predictive model, some regression model, and then use some algorithm that finds sequences with high predictions, maybe also accounting for some notion of uncertainty. And this kind of approach that I just described has been used to design everything from small molecules to materials, fluorescent proteins and enzymes, binding proteins. There's a ton more potential applications here. However, the problem here, of course, is that the model, the orange thing, is not the same thing as the black curve, the ground truth. And furthermore, to make everything more annoying, it generally deviates from it the further you move from the training data, which is a problem because the whole point of design is to move away from your training data to find something better, like over here. Okay, so now that we have this dilemma, if I design a protein that the model says is super good, how do I know it's actually really good instead of the model just being very wrong? So in this single shot setting, our work provides a frequentist notion of uncertainty quantification so that you can hedge your bets about which design proteins are actually worth making in the lab. So in particular, given any design sequence X design, our method gives outputs a confidence set C alpha that contains the true label of that design protein or Y designed with guaranteed high probability. And we do this by generalizing a technique called conformal prediction to a kind of special kind of distribution shift that we also introduced in this work called feedback covariate shift. So this is kind of an important idea in the paper. So let's first look at what that is. So feedback covariate shift, or FCS, is a kind of distribution shift where the training and test data are not just from different distributions, but they're actually statistically dependent because the training data was used to choose the distribution of test inputs. So to break that down a bit more, we're going to start with training data, these blue dots. Now we're going to train some predictive model. This can be whatever you want, neural network, Gaussian process thing, boosted trees, whatever you think is appropriate. We're then going to leverage some design algorithm that consults that model in order to propose new inputs. So for example, this could be new proteins that are believed to have high Y values. So this design algorithm, we just think of it as some mapping from the training data to a distribution over X. So it could be kind of anything you care to be doing. It could be training some conditional generative model that leverages these predictions. It could be some crazy genetic algorithm that incorporates all sorts of heuristics that you think are appropriate. It could be some sort of optimization algorithm. Anything that you want to use to have the predictions help you find kind of better points. All right, so this whole pipeline actually means that the training and design data are statistically dependent. And it's this dependence that actually distinguishes feedback covariate shift from other forms of distribution shift that we see a lot in the machine learning literature. And I should note that this dependence is super hard to analyze if I don't assume anything more about the design algorithm or the predictive model, right? If this is some complicated black box neural network and this is some genetic algorithm, there's no way for me to really reason in closed form about what this dependence might look like. So in spite of this dependency, our work generalizes conformal prediction to handle data under feedback covariate shift, meaning we can basically do principled uncertainty quantification for the design data. Okay, and I'll point out this actually happens a lot in machine learning. So feedback covariate shift is more common than just in design. We often want to use a trained model to make decisions, and then those decisions result in some different distribution of data. So active learning, adaptive experimental design, Bayesian optimization, performative prediction, as we heard from Banki and Tiana in recent days. These are all situations where a model is being used to, in some way, influence the next data that it's going to encounter. So our work, which generalizes conformal prediction to feedback covariance shift, actually applies to a lot of these settings as well. Okay, so now I'm just going to give a high-level overview of conformal prediction. In conformal prediction, I have a predictive model F, and the goal is, given a test input, X test, I want to construct a confidence set C alpha that has a property called coverage. That means that it contains the true label Y test with probability at least 1 minus alpha for any user specified alpha. Okay, that's a cool goal. The really cool part is that the conformal approach allows you to do this, accomplish this goal, without any assumptions on the model F or on the true relationship between X and Y. F can be arbitrarily misspecified or not. It's also cool that coverage is actually a finite sample guarantee. So there's no asymptotics here. There's no training data going to infinity for me to get something meaningful. The one thing we need, though, which is the key super important assumption, is that the training and test data need to be exchangeable for the kind of original conformal prediction work. And exchangeability just means that if I shuffle the order of the training and test points, their joint probability doesn't change. So an example you can keep in your head is just IID data. That's a kind of go-to example for exchangeable data, but exchangeability is actually quite more general than IID. However, even though it's more general than IID, it certainly doesn't include designed data. It doesn't include a feedback covariance shift where the training and design data are not just from different distributions, but they're like have this weird dependency. So basically our main technical contribution in this work was figuring out how to generalize the ideas in conformal prediction beyond exchangeable data to get coverage under feedback covariance shift. So this means that we can construct confidence sets for design proteins that contain the true label Y test with guaranteed high probability. Okay, so before we wrap up, I want to show a use case for this that I think is particularly neat and promising, and that there are kind of open threads to really figure out how to do well, which is how our method can be used to guide design algorithm selection. So the goal in this example I'm going to walk through is to design fluorescent proteins at both a blue wavelength and a red wavelength. And my goal is to get the fluorescence to be brighter. So we use data from a paper that measured the blue and red brightnesses of a complete landscape of protein variants. We then simulated protein design experiments by randomly sampling some of that data to serve as training data, training a regression model, leveraging a design algorithm to design new proteins, and then using our method to construct confidence sets for those design proteins. In particular, the design algorithm was to sample sequences X from this distribution, P of X. So P of X is just an energy-based model where the log likelihood is just proportional to the model's prediction. So in any words, for any sequence X, the higher the predicted brightness, the higher the likelihood that we choose that sequence and call it our design sequence. So I'm going to note that in this design algorithm, as with many design algorithms, there's an important hyperparameter called lambda, or the inverse temperature. And this basically controls how aggressively we want to climb the predictions of the regression model. So greater values of lambda will lead to design sequences that tend to have higher predicted brightness. So I'm going to show you shortly how you can use our method to set a value of lambda in a more informed way. But first, I want to go back to coverage briefly, as this is a key theoretical property that we focused on. So the y-axis here is empirical coverage, and that's the fraction of trials of this simulated experiment where our confidence sets did indeed contain the true label of the design protein. And the x-axis is different values of lambda. So here we want a coverage level of 0.9. That's this dash horizontal line here. And you can see that for a wide range of inverse temperature values and amounts of training data, indeed we get empirical coverage above 0.9. And the nice thing is that it's also not like too much over 0.9, which means it's not being kind of overly conservative. All right, so let's return to this question. How should we choose lambda? We have a predicament because greater lambda means higher predicted brightness, which we like, but it also means at some point higher predictive uncertainty, because at some point your predictions get so high it's bigger than anything you've seen in the training data, and so you wonder like, okay, is the model just kind of going crazy here? So these two things kind of trade up. How should we set lambda? So we can actually use confidence interval width to visualize and navigate the trade-off between these two things to set value lambda. So I have a plot like this where the x-axis is the average predicted brightness of the designed proteins. The y-axis is the average confidence interval width. So on the x-axis, I want this to be high, like to the right. And on the y-axis, I want this to be small. I want low uncertainty in my predictions. So we have two curves here. The blue one is for blue fluorescence, the red is for red fluorescence. And each point on the line corresponds to a different setting of lambda. So the first thing to note is that for both, there's this trade-off. For any unit increase in average predicted brightness, I have to pay for it by being more uncertain about those predictions. So we see that trade-off for both, but qualitatively, we can also see that the trade-off appears a bit different for the two. The slope is much less steep for the blue fluorescence than the red. And you can kind of explain this by, this could be attributed to things like, oh, maybe the assay for the red brightness was noisier, or the model was better specified for the blue fluorescence. In any case, you kind of see this different qualitative behavior and trade-off. And so this kind of point can allow us to select a hyperparameter in an informed way. So for blue, for example, I might be willing to pick lambda equals 6 because I can get a pretty high average predicted fitness while not paying so much in increased uncertainty. Whereas for the red fluorescence, I might only want to pick like lambda equals 2 because if I go any higher, I keep having to pay like more uncertainty for those predictions. As an example. Right, so as a high-level summary of this last part of the talk, in this work, we introduced feedback covariate shift. We generalize conformal prediction to that kind of distribution shift that has dependencies between train and test data. And this basically enables us to do uncertainty quantification for design, and in theory for any design algorithm using any kind of predictive model. And so, again, I think this direction of using this kind of method for design algorithm selection is quite interesting. I'm almost done.
There's a question.
Yes, we're almost there. Almost there, way more. You want to get down? So back to this question. I hope I've convinced you that we can make steps toward using machine learning in the process of science in a reliable way. And I'm really excited about extending this line of work. There's a lot of open challenges here, and I think tackling them will be really important for enabling machine learning to accelerate the process of science in a way that's actually reliable and responsible. Okay, I want to end with this awesome explicit I found about confidence intervals. So hopefully mine are more